Alright, so carrying on from the lesson before, we spoke about the different tests that are used to identify reducing sugars. Now, reducing sugars is a type of carbohydrates, so we can actually carry out the Benedict's test as mentioned yesterday. Okay, and in this Benedict's test, we will use the reagent, right, Benedict's solution, and we will use equal volume of Benedict's solution with equal volume of sample. And then after shaking the mixture, we will place it in a boiling water bath for 2 to 3 minutes. And then we will be able to observe for a color change. Now, all these color changes are not quantitative. That means they are qualitative tests. They only allow us to see whether a particular nutrient is present by identifying its presence by changing color. Now, if you change a color, you do not know how much there is. Okay, so basically we have two possible outcomes. One, if let's say the Benedict solution remains blue, that means reducing sugar is not present. If by any chance there is a change in color, that means reducing sugar, which is a type of carbohydrate, is present. We also saw the different range of colors, so this indicates that reducing sugar is absent, and any color change indicates that reducing sugar is present. So why do you have different color changes? That's because you have different amounts of reducing sugar. Now, although Miss Lee did mention that this one cannot tell you how much it is, but these are all estimates. Okay, so you may want to write this down in your notes that this is an estimate. Okay, if you have brick red, it means the most amount of reducing sugar. If you have the green, that means it's the least. If you have blue, that means it's a negative test. There is no reducing sugar present at all. Alright, so today we're actually going to be talking about the different types of tests for the other types of nutrients. Now, another type of carbohydrate is your starch, and you know that starch is actually a complex carbohydrate. So the reducing sugar test that we saw just now is actually single sugars, right? They are the simple sugars. Now we want to talk about complex sugars, that means your carbohydrates. Okay, now in your carbohydrates, we carry out a test that uses iodine solution. That's why the term iodine test. Now, before I actually take you through the steps, alright, I would like you to watch this simple video to see how the iodine test is being carried out. Okay, this is the iodine test, and the iodine test is used to test for a sample for the presence of starch. In this iodine test, we will perform this using the reagent iodine solution. And we will use starch solution, but on the day of the exam, you may be given an unknown specimen or sample. Okay, so take a plastic pipette and drop 1 drop of the starch solution or your unknown sample onto a white towel. To the same sample, add 1 to 2 drops of iodine solution. Observe that the iodine solution turns blue-black in the presence of starch. Now, onto the same towel, we will drop 1 to 2 drops of iodine solution, and you will notice that the color of iodine solution is yellowish brown. So you can see that a negative iodine test, in the absence of starch, iodine solution will remain yellow-brown. Okay, so I hope that you actually got some idea of why we carry out an iodine test. So the steps of the iodine test is, you will notice that we actually drop a few drops of iodine solution to a liquid solution on the white towel. In this case, we are already using a known solution that contains starch. However, sometimes in the practical, you may be given a solution that you do not know what is that particular solution. Now, to that same solution, we will add a few drops of iodine solution. Now, you may want to take this time to fill in your notes first, so you may want to pause the video here. Alright, so you notice from the video that if starch is actually present, alright, so I'm just going to draw your attention to it. If starch is present, that means it is this blue-black solution that we see here, you will notice that your iodine solution, alright, actually turns blue-black. Now that's because there is a chemical reaction that allows iodine to turn blue-black when it is in combination with starch. Alright. Okay, you will notice that the iodine solution actually turns blue-black in the presence of starch. However, now if you look at this little yellowish spot here on the white tile, this actually only contained iodine solution. We did not add in the starch solution at all. So that means when starch is absent, that means starch is not around, your iodine solution will stay at its original color, which is yellowish brown. Alright? So you may want to take some time to write down and fill in the blanks in your notes. Alright. Okay, so the next test that we're going to be looking out is actually the test for fats. Now, if you remember, your fats is actually made up of one glycerol molecule that is attached to 3 fatty acid chains. Now, these 3 fatty acid chains and the glycerol molecule are chemically bonded together. So how do we test for fats? We'll watch the video first and then we will fill in our notes, okay? Okay, this is the test known as the ethanol emulsion test, which is to test for the presence of fat. To a clean dry test tube, add 2 cm cube of oil. Add 2 cm cube of ethanol to the same test tube. Shake the mixture well. Add 2 cm cube of water to the same test tube. Observe what you will see. If fats are present, you will get this cloudy white emulsion. If fats are not present, you will get a clear solution. Okay, so from the experiment itself in the video, you would have noticed that we will actually carry out an ethanol emulsion test. Now, why do we use the word ethanol emulsion test? Because we actually use the alcohol, ethanol. Okay, and why do we say emulsion? Because from the video you can tell over here, okay, that if fats are present, you will get this cloudy white mixture, right? Now, this is actually known as an emulsion because fats and alcohol cannot basically because fats and water, I apologize, fat and water are basically immiscible. That means fat cannot dissolve in water. So what do? We will add 2 cm cube of ethanol to 2 cm cube of the sample. We will shake the mixture vigorously in order for you to shake it well. And then after that, we will add 3 cm cube of water to the test tube. And if fats are present, just like this video over here, you will notice that you will get a cloudy white emulsion. Okay, so the last video that we're going to be watching and the last experiment that we will actually be doing in the lab is how do we identify for the presence of proteins. Now, can you recall earlier in the lesson, we said that proteins are actually made up of the building blocks, amino acids. So basically, different types of amino acids represented here by the different shapes, okay? The different shapes here will be bonded together by your peptide bonds. Okay, why do we use peptide? Because of the word P, protein. Okay, so just like here. Now, these bonds are peptide bonds. So these peptide bonds will join all the amino acids to form a polypeptide, okay, which is a long chain. And then many polypeptides will then coil around each other to give you a protein molecule. Okay, so now before we actually look at the steps again, let us identify what the steps are by watching this very, very simple short video. The next test that we are going to conduct is the burette test using the burette reagent. This test is to test for the presence of protein. For the purpose of this test, we will have two samples, an egg white solution which contains protein and glucose solution which does not contain protein. Add 2 cm³ of the egg white solution to a clean test tube. To this test tube, add the burette reagent drop by drop, shaking after each drop, and observe for the color change. You will notice that if protein is present, the blue burette reagent has turned into a purple color. So add 2 cm³ of glucose solution to a clean test tube. Add burette reagent drop by drop, shaking after each drop. Difference in color. The glucose solution does not contain protein and it will not turn purple, unlike the egg white solution that contains protein. Okay, so over here you can tell that we had two solutions given to us, one that contains egg white and the other one that contains glucose. Now you know that glucose is a carbohydrate, so it does not contain protein, whereas egg white actually contains protein. So the type of solution that we actually use in this experiment is known as a burette solution. That's why the name of the test is burette test. So we will add 2 cm³ of this burette solution that you can see here, which is blue in color, to a solution in a test tube. We will shake the mixture well, okay, to mix everything together. Now, if proteins are present, just like this test tube over here, you will notice that the color changes from blue to violet. Okay, you may also use another term in English known as lilac. Alright, lilac and violet, or you can even use the word purple. Okay, it's a very, very nice, beautiful color. Now, you would notice that in the second test tube that Miss Lee is labeling here as number 2, we use a glucose solution which does not contain protein. So, if protein is absent, you will notice that the solution actually remains blue because the original solution, the original burette solution here was blue in color. So, as long as it remains the original color, we say that that particular nutrient is not present. Alright? Okay, so this is a very, very important table. I will give you 1 minute to have a look at this table. Alright, you may pause the video here just in case you need to copy down and write down certain things. Now, we have actually tested, or we have actually seen 4 different tests that allow us to test for the 3 different types of nutrients that we've had. Okay, one is your reducing sugar test where we use the Benedict solution. One is to test for starch, which is to use iodine. Now, reducing sugar and starch are all under carbohydrates. Okay, so we have one test for carbohydrates, reducing sugar, which is usually used to test for the presence of glucose, which is a simple sugar or a single sugar. Okay, there's only one single sugar. Starch, which is a complex or very complicated carbohydrates because you have many glucose joined together. Okay, then the next type of nutrient that we actually saw was to test for the presence of protein, which we use the burette solution. Alright, and then the last test that we actually carried out, which is the test to test for fats using the ethanol emulsion test. Alright, now it is very important that you take note of this. That means you must be able to identify what are the results, okay, for a positive as well as a negative reaction. Now, this is a very common kind of question that may come out. Okay, so you will need to be able to complete the worksheet that has been attached for you, and you must be able to complete all the MCQ questions, because most of the MCQ questions will usually be asking you, oh, you know, if reducing sugar is not present, what would the color of your Benedict solution be? If starch is present, what is the color of your iodine solution? So it's very important for you to remember this table and commit it to memory. Alright, so this brings us to the end of our online lecture. You may now carry on with the worksheet, okay, that is inside, that has been attached inside your home-based learning, and then the answers. Please make sure that you take a picture of your work and submit it to me via your email, okay, so that I can check whether you are on the right track. Okay, thank you.